Welcome to our series, Answers That You Can Get in 1 Minute or Less in iPathway Guide. Today we will answer two questions that are important in any experiment. These are, what are the most affected biological processes in my phenotype, and how can I eliminate or reduce the false positives? In a previous video, we showed you that the typical approach used by most software platforms for gene ontology analysis results in over 100 significant Go terms, most of which are false positives. Here, we will show you how to weed out these uninformative results and identify the most truly affected biological processes in your phenotype. To illustrate this, we will identify the most impacted biological processes from a public data set where the authors made two important discoveries: that p53 was positively regulated and that cell proliferation was inhibited. In iPathway Guide, this analysis is available under the Gene Ontology tab. Using the standard Bonferroni correction for multiple comparisons yields hundreds of uninformative results for you to wade through. In iPathway Guide, however, you can select high specificity pruning in the p-value corrections drop-down menu. Using this proprietary approach, the analysis now reports only 3 significant biological processes. The very first one pinpoints the involvement of p53, which was the very title of the paper describing this work. The second process mentioned in the abstract, negative regulation of cell proliferation, is also one of the top 3. We have now eliminated hundreds of false positives, leaving more time to devote to better understanding the data. Using iPathway Guide, you'll stop wasting time on false positives and quickly get to the heart of your work.